Apis mellifera selects flowers based on floral morphology. Emma McFarlane, Entomology 320. Apis mellifera, a honeybee, is constantly pollinating all flowering plants. They are important for reproduction of immobile flowers. Flower morphology encompasses a broad range of features, including the number and shape of petals, the number of stamens, the size of the petals, and the arrangement of the ovaries and styles. Everything considered by bees when selecting flowers. Human and honeybees both have 3 photoreceptors and are trichromatic. However, honeybee vision is based on green, blue, and ultraviolet light. Their vision impacts what petal they go to. What is Apis mellifera floral preference in terms of morphology? How do the sex phases of flowers influence the foraging behaviors of Apis mellifera in terms of inflorescence choice and time spent foraging? The objective of the research is to understand the sex phases of lavender flowers affect honeybees' choices and foraging behaviors, focusing on the preference for certain flowers based on nectar and pollen availability. There are 2 key findings. One was inflorescence choice. Bees foraging for nectar prefer inflorescences with more female flowers due to the higher nectar volumes. Bees seeking pollen and nectar opt for inflorescences with a balanced number of male and female flowers. 2. Time spent foraging. Nectar-only foragers spend more time on inflorescences with more female flowers. Pollen and nectar foragers spend more time on inflorescences with more male flowers necessary for pollen collection. Figure 2 of that research demonstrates the influence of flower morphology on Apis mellifera foraging. Because female flowers have more nectar, nectar-only foragers give them priority and spend more time tending to them. Because of their need for resources, pollen and nectar foragers are attracted to the male flowers since they produce pollen. This emphasizes how crucial flower sex is to bee foraging decisions and how they tailor their foraging according to floral traits. How do flower sizes affect the preference on Apis mellifera? This research objective is to examine how Apis mellifera preference and behavior related to foraging are affected by blossoms, sizes, paying attention to visual cues, and foraging efficiency. There are 4 key findings. Visual attraction, because they are more visible and appear to have more resources, larger flowers drawing more bees. Pollination efficiency, larger blooms, which frequently offer easier access to greater amounts of nectar and pollen, are better pollinated by Apis mellifera. Selective preference. Larger flowers are continuously preferred by bees, probably because they provide greater rewards and require less energy to forage. Learning and memory. Over time, bees become more efficient at foraging because they have tendency to retain the locations of larger blossoms and give them priority on subsequent foraging visits. Why does Apis mellifera use flower pigment patterns in foraging choices? The objective of the research is to learn how Apis mellifera bases its foraging decisions on the pigment patterns in flowers. There are 4 key findings. 1. Pattern use in foraging. Bees have the ability to employ intricate foraging strategies by using the pigment patterns in flowers to differentiate between flower morphs that offer varying caloric rewards. 2. Energy maximization strategy. Bees favor flower morphs that provide more calories, which is consistent with the energy maximization model. 3. Learning and decision making. After visiting a single, less satisfying flower, bees frequently modify their preference in response to changes in caloric rewards. 4. Pattern complexity and errors. Bees exhibit a higher error rate in flower choices when navigating between flowers with pattern differences compared to color differences alone. The slide is part of figure 1 of that research, and A is a bilateral pattern. The flower is divided into two equal halves, one side painted blue and the other white. B is a radial pattern. This pattern features a blue center circle surrounded by white. C is a radial pattern with white center. This pattern has a white center circle surrounded by blue. Figure 1 in the document shows different flower pigment patterns used in experiments to observe Apis mellifera foraging behaviors. The results indicate that bees can distinguish these patterns and use them to choose flowers, offering higher caloric rewards, effectively utilizing visual cues to optimize their foraging decisions. Conclusion. Influence of flower sex phases on foraging behavior. Apis mellifera maximizes resource intake by displaying a definite preference for female flowers that are high in nectar. Bees effectively manage their energy use by focusing more on their foraging effort on male blooms for pollen and female blossoms for nectar. Effect of flower size on bee preferences. Because larger blooms are easier to see and provide more substantial resources, Apis mellifera are drawn to them, which improves pollination efficiency. Recalling and going back to the bigger blossoms, bees exhibit adaptive behavior that optimizes obtaining resources. Role of flower pigment patterns in foraging resources. Bees maximize their energy by identifying and choosing flowers with higher caloric rewards based on patterns in the floral pigments. Bees learn to deal with complicated patterns more slowly and make more mistakes when compared to basic color distinctions, even though they can quickly adjust to the changes in rewards. The study emphasizes how important floral morphology is to bee feeding strategies, and it affects pollination ecology in important ways. It demonstrates how knowledge of floral characteristics may direct farming operations, improve pollinator efficiency, and plant reproductive success. By focusing on floral morphology in crop and garden planning, the study offers practical insights that could improve pollination strategies and agriculture productivity. The flower doesn't dream of the bee, it blossoms and the bee comes. Mark Neckbo.